Okay. All right. So my name's Kathleen. My teammate is Weina. We are from Hammerne's Medical Image Analysis Group at SFU in Vancouver, and our mentor was Miguel from Mojo Diagnostics. Sorry, that was in the way. We labeled this talk Annotations more than just labels. So what do I mean by that? Why is this important to everyone? We rely on annotations to train our supervised learning methods. And because of this, the quality of our methods rely on the quality of the annotation as well. And we all face technical challenges related to annotations. For instance, how can we actually ensure that the features are correctly recognized? And how do we even know if we have noisy annotations in our data set? So considering the overall theme of the hackathon, bridging the gap from method to clinic, here's sort of an overall pipeline of a typical medical image interpretation process in a clinical setting. So the radiologist needs to perform pattern recognition to identify certain image features and then somehow connect them back to their medical knowledge and their expertise to come up with a diagnostic hypothesis. So for us, we believe that utilizing abundant medical data is imperative to bridging this gap, but the cost of labeling data is still very, very high. So one possible improvement to this pipeline is to introduce novice physicians or annotators into it. But one thing to note is that clinical reasoning would probably hardly be outsourced because of the domain experience that is required. However, one potential improvement is that we outsource this pattern recognition part. And this is where annotators and labeling comes into play. So outsourcing clinical problems to laypersons for annotation generally takes this form. Given medical images, you perform pattern recognition to come up with some sort of image features that you then base a conclusion on given simple rules or protocols that are provided to the annotator. Some example problems are biomedical image tasks such as microscopic cell counting and cell morphology recognition, which we'll sort of try to tackle in this presentation, or clinical image tasks like diagnosing a skin lesion, say as melanoma, or applying a 7-point checklist to it. So task 5 specifically was increasing the consistency and quality of annotations. What we did is we came up with a couple of scenarios. One is the first case is that you already have data collected and you're concerned about the quality of the annotations of that data set. And case 2 is you're looking to collect data and you want to ensure the quality, and maybe you're looking for ways to scale up the annotation quantity and quality. What we propose is sort of like a toolkit, if you will, or that was like our grand idea, a set of tools that help both machine learning and clinical practitioners understand data annotations, which we just call Annotation Insights. And we'll just kind of walk through a proof of concept on the data set provided by our mentor. So a real world problem is that male fertility rates are potentially declining. Infertility may range between 10 to 25% of the entire population. And an issue is that a lot of these people don't have a clear-cut diagnosis to the cause of their infertility. And this is because it may be expensive to get such a diagnosis. You may not have access to a specialist. And also because there's complex morphological defects to sperm cells that could cause infertility. So the data that was provided by our mentor is a spermatozoid data set. And in relation to task 5, this is an annotated data set of motile sperm cells. They were imaged using microscopy and labeled for morphological defects according to the World Health Organization protocol. And annotators, to our understanding, were freelancers. They were pre-screened and selected. And the question, the real world problem question, is how good were they? So first, we just looked into a breakdown of the annotation in the classes. There were 4 classes in one of the data sets that we looked into. 3 are defect classes and a normal class. There were 3 unique annotators. Each sample was assigned to only 1 annotator. And what we saw was that the class samples were roughly balanced amongst the annotators, which was good. Here's a schematic of abnormal forms of human spermatozoids from the World Health Organization website. The data only is concerned with head defects. So here there's 3 classes of defects, and these are actual images taken from the data set provided, as well as a normal class. So one of our proposed solutions was to extract features from these images using pre-trained convolutional neural networks. We are not doing any learning in this case. We're simply taking something off the shelf and trying to get some sort of features that we hope capture the underlying morphology of the cell. We then apply some dimensionality reduction to these features to hope to understand the manifold of the data better, and then try to detect outliers based on visualizations of the data. So here we have principal component analysis applied to features that were extracted from a pre-trained network. And what we're trying to do is see, one, do they actually cluster? So without learning involved, are their clusters representative? And then are there outliers in these clusters? So here I've kind of tagged some that may be outliers of a particular class. One thing to note is that dbScan was unable to cluster the data. This may be because the data set was pretty small. It was 216 samples across 4 classes, but nonetheless, it's neat, cool data, and we just wanted to apply a proof of concept to it. So if we look at the data on the right and the visualization in 2D space using principal component analysis, and then focus only on the normal classes, we would go back into these and see who annotated these samples, what do these samples look like, and try to gain some sort of insights into not only the features, but the annotators. And we can do that across all 4 classes of the data, just sort of going in, seeing where data falls and who annotates the data. So key takeaways in this case, where we already have a data set collected, one of our solutions is to detect outliers by a clustering method. But some things that we note is that the differences between morphological defects or classes in this case may be too subtle for freelance annotators. And a whole image label may not suffice in capturing features. So in the second case, so if you're looking at collecting data in the future, some things that we propose as solutions is enhanced annotation. So maybe incorporating uncertainty into the label. So as opposed to a single annotator just classifying it, binning it into a category, you could incorporate some sort of uncertainty. And second, an intention mechanism, say for instance, gaze tracking might help to understand visual cues in the data. And difficulty of the sample. So here on the bottom, I just kind of played around with MediaPipe, which I believe is open source and maybe from Google. And one idea is to potentially track the gaze of an annotator and see how many times they have to reference a protocol for a given sample, which may indicate how challenging that sample is to annotate, and also whether the annotator is, say, fatigued or interested in the task. Okay. Finally, a huge thank you to all who supported this fun event. It was great. The organizers, mentors, keynote speakers, and also the institutions. Thank you very much.